Welcome everyone. Thanks for logging in today. So today's speaker is Professor Giovanni Valenti from the University of Bologna, and he's going to discuss electrochemiluminescence-based biosensors. And it's a real pleasure to have you here today, Giovanni, and I look forward to your talk, and we'll have questions afterwards. So over to you.
Okay, so thank you very much. First of all, thank you very much for organizing this nice series of Congress. I really like this. So today my talk will be focused on the activity that I'm doing since my master degree, more or less. So since my activity is focused, well, I have different field, but one of the most interesting field I think is on electrochemiluminescence. So I think ACL, so what I call ACL, electrochemiluminescence, is a nice idea, is a nice research activity where you can merge the fundamental research to the real application, so to the real diagnostic application. So the title of my presentation is Electrochemiluminescence from academic curiosity to an industrial success. And in fact, I will range from the molecular point of view, so from the mechanism of the signal generation to the real application. So first of all, let's move on what ACL is. ACL is, I'm sorry for the people in the audience that already know this, but ACL is an electrochemically induced chemiluminescence. So means you apply a potential and you generate light. Here you can see, here you can see in a working electrode during the ACL. You can see that only the emission occurs only from the working electrode. So electrochemically induced emission. That's an amazing idea. Well, the guy that invented the techniques, the father of this technique is Professor Alain Bard, that all you know, of course, and he developed ACL in water for the first time, and I will go through this invention. So there are two main books if you are interested. There are many reviews here. I just list my favorite one. So my favorite reviews, but there are two main books, one from Alain Bard, and that's quite old book, and another one very recently in 2020, from Neshi Sojic group. So how we can generate light with electrochemistry? The main idea is to, so the main mechanism involves a reduction process, so generation of radical cation and generation of radical anion. So when you can oxidize one species, one dye, you can reduce this dye, and both are in the diffusion layer and can generate the excited state that emit light. This can occur between two electrodes or at the same electrode surface. How it could be generated the same electrode surface, switching the potential between the first oxidation and between the first reduction. If the electrode is the same electrode, we call in the field, we call annihilation mode. And here you can see this nice blue color emitting from an acetonitride. In fact, this mechanism was observed, but this is another story, very early in 1927 by Dufford, that was doing electrolysis in a protic solvent, and he observed blue emission in the middle of the two electrodes during the electrolysis. So annihilation, switching the potential, go to negative potential, generate the radical, opposite potential, we move to oxidation potential, we generate the radical again. Both are in the diffusion layer. Here I schematized the diffusion layer. I don't know if you can see this, but both are generated at the same electrode in the diffusion layer. And so the two concentration profile are overlap, and you can generate the excited state that after emit and generate the light. So a spike of light. And you can again switch the potential in the negative potential and again in the positive, and you generate light in each in each potential step. Okay, amazing. They demonstrate in the first, well, 2000 more or less, they demonstrate that they can generate ACL in this condition. However, in this way, you must need very aprotic solvent. So a solvent that allow you to go to a very quite negative and quite positive. Typically, this solvent is acetonitrile. Here I take this very nice picture from Francis in this very nice work. And as you can see clearly here, you have to move to the oxidation potential that is around 1 volts and to the very negative potential that is around minus 1.5. So you need very aprotic condition, and you can do that only, mainly only in acetonitrile or in THF, for example. So you need non-aqueous media, otherwise you will have the hydrogen evolution reaction, of course. And of course, doing that in non-aqueous media, you cannot have bioanalytical application of this. Why we would like to work on in aqueous media? Why? That's very important, because in ACL, as an bioanalytical technique, if you think, you have a great advantage of the chemiluminescence, so very high signal-to-noise ratio, because you don't have excitation source, you don't have light scattering from the excitation source. This allows you to have very low background and very good signal-to-noise ratio. This is also due to the fact that the emission is localized, of course, from the electrosurface, so is localized only on the electrosurface, and the potential control the emission. So you can decide when to apply the potential and with a very high signal-to-noise ratio. This allows us to go to very sensitive, ultra-sensitive quantification of many biomolecule, proteins, hormone, etc. So there are two main, well, as far as I know, there are two main industrial application of this. The first one developed by Roche and the Hitachi technology, and is based on microbeads in both of these cases, but in this case you have a sandwich immunoassay, so you have your capture antibody, your analyte, and your detection antibody, that is this red antibody labeled with an ACL dye, so something that emits upon the potential application. So the combination of this kind of technology with microbeads is very important and very useful in the field, because you can have magnetic separation of the beads. You can use magnetic field to attract magnetic beads on the electrode. On the magnetic beads, you have the sandwich assay, and after you can generate the light by simply apply the potential. This other application is from mesoscale, and also in this case, they use a different approach. So it's a multi-rate plate, but they still have this sandwich antibody on the electrode. Well, these are the two instruments. The first one is called Alexis, and the second one is called Sector. So these two instruments have a great field of application, in particular for the diagnosis of markers, tumor markers. Okay, so here, if, for example, the Alexis technology, so the Roche one, you can have more than 150 biomolecule. You can quantify more than 150 biomolecule, including anti-SARS-CoV-2 antibodies. So it's also a very actual situation. And this is considered one of the top-selling diagnostic system from Roche Diagnostics. So it's very interesting. What is also interesting that this kind of combination between microbeads and ACL was initially invented by Hygen International. So this is, I suggest you to read about this combination between a small biotechnology company that is called Hygen International. They were the one invented the combination between microbeads and ACL in the 1991, and after they moved to Roche. So it's a very, very interesting industrial application. I cannot move more. Okay, so how we can generate the light? I don't know why I have all this arrow in my screen, but it's okay. How we can generate light in aqueous solution and without annihilation, because the potential windows in water is too small for the annihilation method that I showed before. We can generate light with these two guys. So basically, ruthenium trace bipyridine is one of the main ACL dye. So is the main ACL dye, and it was the one developed for the first time by Barton co-workers and is still used. So if you go to the hospital now to screen some tumor markers, if you go to the hospital, they are still using this ACL dye, ruthenium trace bipyridine. So is our star in the field. The other star in the field is a coreactant. So another guy, another species, another chemical species called tripropylamine, TPA, is an amine and is typically a sacrificial coreactant. So it's a large amount and is sacrificial coreactant. So what occurs at the electrosurface? You can see here, I schematized the immunoassay system, the detection antibody labeled with our ruthenium trace bipyridine complex, capture antibody, analyze everything on the electrosurface. You can have this on the electrosurface or on a beads. That's two different approaches, but the same mechanism. And coreactant. So you can oxidize the coreactant, you can generate radical cation, and this is unstable oxidation. This is unstable radical cation, and you can have a classical electrochemical chemical mechanism where you are degrading the radical cation to the radical, and you have both of these in the diffusion layer. This, of course, strongly depends from the time life of the radical cation, and you have a guy that can be reduce your ruthenium trace bipyridine, a guy that can oxidize your ruthenium trace bipyridine, and you can generate excited states. That's amazing, because with the easy and simple potential step, just applying the oxidation potential for the TPA, just applying the positive potential for the TPA oxidation, you can generate the emission. And this potential is typically 1.1, 1.2, so the standard potential for TPA is 0.9. So you have sacrificial coreactant and catalytic cycle. This, of course, strongly depends from the TPA radical lifetime. So the radical lifetime really affect the emission, really affect the emission at different distance from the electrosurface. This is a catalytic cycle. Okay, so this is applied in both the system that I showed before. So it's not only mechanistic studies developed by Bard and co-workers in the 2002, but also a real commercial application. In the case of beads assay, you have the emission from beads, but you oxidize the TPA. In the case of multi-array technologies, you have the sandwich immunoassay onto the electrosurface and you oxidize the TPA. Okay, so one of the key points, as I told you before, is the optimization of the coreactant distribution from the electrosurface towards the bulk. Here I take this a very nice plot from Sojic paper. So you can see here, this is the electrosurface, this is the concentration of the radical. So sorry, this is the distance from the electrosurface towards the bulk of the solution. You see the red signal is the ACL emission. You see we have a maximum around 0.51 micrometer distance from the electrosurface, and after a decay. This maximum depends from the radical lifetime. Okay, so how Bard, back to the Bard paper in 2002, where he demonstrated for the first time this mechanism, how he demonstrated this mechanism. He used an ITO electrode, this is ITO electrode, and he labeled the ITO electrode with our star ruthenium trace bipyridine. And after he used a millimeter electrode with a hemispherical tip, and he moved this onto the ITO, modified ITO, and move this back and towards back and move close or at a higher distance from the emitting ruthenium layer, and it generates TPA radical and radical cation at the tip. In our group, we call this surface emission, beads oxidation, tip oxidation, tip generation, surface emission mode. Dr. Bard put in the 2002, he put a photomultiplier tube under the ITO electrode. So by moving towards the bulk of the solution or close to the electrode, to the ruthenium layer, we can here obtain a map of the emission. And here we have the ITO electrode, and if you move distant from this electrode, you measure this decay of the ACL intensity. This decay is strongly connected with the travel distance, so travel distance of TPA radical and radical cation. So by moving up or down, it can manage to measure this travel distance. And this was really a revolution, let's say, in the field. In fact, from this paper in 2002, after that, it demonstrated that it can occur ACL in water, and we have this exponential increase of these are the publication, but also exponential increase of the commercial application. And we have this exponential increase of the publication. And now in the 2018, it was more than 3,000 papers per year on ACL. So it's something very, very important. And here we come to our research activity. Basically, I divided the presentation in 3 parts. So the first one is on the mechanistic optimization. So we start from the Bard paper, the 2002 paper, and we did again the same experiment, different tip substrate. In fact, we call it tip generation, substrate emission, inspiring by the CCM scanning electrochemical microscopy word, but this is another story. So we did the same experiment, moving the tip closer to the ruthenium layer or far away from the ruthenium layer, but instead than a photomultiplier tube, as Bard did, we used sorry, I don't know why, but I have this. Okay, we use a microscope. So we combine this emission with a microscope, and this is what we call ACL microscopy. This is our tip. And if you can measure the current as a function of the distance, the current, of course, does not change too much, and you can measure the ACL intensity. And here you have an increase of the ACL intensity when you go closer to the electrode, to the ruthenium ITO layer. And we have an increase, and this is our orange points, orange dots. You see that we have an increase that is very similar to the red curve that are the experiment obtained by Bard and co-workers. So orange dot is our experiment, and the red one is the Bard one. But so this is the movie that we obtain by increasing the potential. We go towards the potential. TPA oxidation occurs at the tip, and we see the emission. But if you go more into the detail of this, I don't know how to go. Okay. If you go and you zoom in the emission that we obtain, the map that we obtain thanks to the microscope, we see that the emission occurs, notice, this occurs from the ITO layer is not homogeneous. We have a donut shape, so black dots in the center of the ITO substrate. So emission, but black dots in the center. And this is why, this is very well known in the field of scanning electrochemical microscopy, is due to the hindering of the TPA. So if you have your tip very close to the substrate, you have the hindering of the TPA due to the shape of the tip, And so in the middle, you cannot have a strong emission, homogeneous emission like in the donut shape. Okay, so what we did, we decreased the size of the tip, and we go on a smaller tip, and we perform again the same experiment with ACL microscopy in the bottom view. And in this case, a smaller tip, this was 200, I think 0.5 millimeter tip, and you see we have homogeneous emission from the ITO electrode. Notice that even in this case, although we did not observe that, we still have the hindering of the TPA that is, of course, cannot be avoided. But if we measure the emission as a function of the distance, like the same experiment that Bart did, we have the orange dot is with a bigger tip. When we go to a smaller tip, we have the blue curve, and we see that at large distance, we have the same trend, but where we go closer to the electrode surface, sorry, closer to the ruthenium ITO electrode, we have an exponential increase. Amazing. We observe this exponential increase only with a smaller tip. Amazing. This means that if we concentrate more, if we concentrate the ruthenium dye, the ACL emitting dye and at this distance, less than 1 micrometer, this is at the distance. So if you concentrate the ruthenium dye in the first micrometer, we can have a higher intensity compared with the long distance. And that's what exactly we did. In fact, if you take the Roche system, so if you take Lexis, typically they use 2.8 micrometer beads, so a bigger beads with the 2.8 micrometer as a diameter. And in fact, if we go to a smaller diameter, we can concentrate the amount of ruthenium dye for our catalytic cycle and in the area where the ACL mechanism is more active, so where we have this exponential trend. And that's what we did. We studied different bead dimension, 2.8, 1 micrometer. We studied many different beads dimension, and we measure it from the top view, so by our microscope in the top view configuration. We call this substrate generation because we are oxidizing TPA at the electron and bead emission mode. And if we go to analyze, to quantify the emission, here I don't have time to go into detail of this, but if you go to the quantification of beads, of ACL efficiency for the 2.83 micrometer beads commercially used, we have this emission, and after we go to this exponential increase. So if we concentrate for a smaller beads like 0.3 or 0.5, with 0.3, for example, we can have an increase of the signal of 8 times, because in that area, in that region, the ACL is more active, is the more active part of the emission. Okay, so as I showed you before, this is the mechanism proposed by Bart and co-worker in his paper, and in order to deep understand what was the mechanism and why we have this discrepancy in the close proximity of the electrode surface, we measure the mass, we apply a potential for a long time, so we perform a long prolongated electrolysis, and we use a classical radical trap to trap the radical electrogenerated. Of course, we measure the radical generated from the amine, so in the alpha position, but we also observe another radical that is this kind of radical come from the CN cleavage of the electrode. That's very in line with our results, because this radical, this time life of this radical already measured by Bart and co-workers is around 700 microsecond. But if we have higher intensity in a close proximity, we should have a radical more reactive with a smaller lifetime. And in fact, if we go to the linearization of this curve, by measuring the lifetime, the traveling distance, what Bart called traveling distance. We measure the 700 microsecond, that is this mechanism, but also a short traveling distance that is, we think, and we have the proof from our analysis, that is the generation of the radical after CN cleavage of the amine. So we have two different pathways. One is for long distance, one is for involve deprotonation, and another one that involve a generation of a shorter lifetime radical, but more active. So let's move to the optimization. So now that we have this concept, let's move to this optimization. So we add to the classical TPA solution, so classical tripropylamine solution, an amine with a methyl group in the alpha position. This amine with a methyl group in the alpha position stabilize this. We put an amine on the, a methyl group in the alpha position in order to stabilize this pathway, to stabilize and highlight the pathway very efficient in the short distance. So we mix and we use this as an additive, and we mix with the TPA, and we come back to our surface generation beads emission mode. And here you can see the beads from with the additive, with this different amine, and the beads without this different amine, and you see that we have an increase of 25%. So it means we are enhancing the mechanism more efficient in the close proximity of the electrosurface. Okay, let's move to the real application, because our idea is to optimize the mechanism for a real application. This is a measure experiments obtaining with Alexis, which was 5 different analytes, and with or without our amine as additive, we have a strong increase of the signal, around 30% of the signal, thanks to the optimization of the mechanism. In this case, we use exactly the commercially available instrument, and we use a collective bead, so multi-beads layer on the electrosurface. Okay, so as I told you before, we call this ACL microscopy, and this brings me to the second part of this presentation, that is a collaboration with Professor Sojic and Andrea Lesch, where we use the idea is to use this kind of ACL combined with a microscope for visualize biological objects. That's very interesting, a new application of ACL, and that can be combined very good signal to noise ratio with a map of biological objects such as cells. This story starts a few years ago, where we started to visualize microbeads, something as I showed you before, for the surface generation beads emission mode. And this starts with Simone Zanarini in this paper. He used ITO electrode for visualize microbeads. Those are big, huge beads. Now we go to very, very smaller beads. But what I would like to show you is that also the surface is a key point in ACL application. In fact, we need to apply for generate ACL. We need to apply 1.1, 1.2 volts for the TPA oxidation, but TPA oxidation strongly depends from the electrode surface. The kinetic of oxidation strongly depends from the electrode surface. And here, we optimize the electrosurface moving from the ITO that I showed you in the slide before towards carbon nanotubes. Carbon nanotubes has a very higher emission compared with the ITO, because the kinetic of TPA is faster, and so you can generate more radicals, and you can have, these are single beads emission. The red dot, the red line is ITO, and the black curve is the emission from the carbon nanotubes film. That's very important, because you must optimize, if you want to work in the ACL field, you must optimize the working electrode material, because strongly depend the emission that you can measure from the electrode material. As I told you before, we moved to biological objects, what I call biological objects. We moved on the cells, and we tested different kind of cells in a collaboration with Neso Sojic. We tested biotinylated, labeled cells with labeled with ruthenium or with antibody, and we grow these cells on electrodes. So it's a carbon nanotubes electrode. There was a presentation a few months ago during August, I think, July or August, by Andrea Lesch about this carbon nanotubes. So he is a master in the preparation of this inject carbon nanotubes electrode, and he prepared for us, and we grow the cells on this. And these are the ACL imaging from cells. So you can see the green one. This is a very nice picture from Bordeaux. The green one is the photoluminescence image. The red one is the electrochemiluminescence image, and we have the combination between the two. What is very nice that we can visualize for the first time cells, and we can visualize cells grow on the electrosurface. But what is the problem? We have this black hole. You can see here the emission, although the emission in the PL, the photoluminescence, is homogeneous. In the case of electrochemiluminescence, is not homogeneous. So these are the main drawbacks. But why? Let's go more in detail into this. So why we have the emission only from the periphery of the cells? Because we generate the TPA radical, and the electrosurface TPA radical need the protonation and need to go, TPA radical need the protonation, and this TPA radical has a time life that in this case was reported here 0.2 millisecond, but as a time life, and so you cannot visualize all the cells. You can visualize only the border of these cells, because the TPA radical cannot reach the very top of the cells. And this was also, we always try to combine in the different, with also different field of application, but we always try to combine ACL and ACL microscopy with a finite element simulation. So these are console simulation, and you see here you have the radical cation concentration profile and the radical concentration profile, and in this case you can visualize this red border are the cell border, so you can visualize until where you can reach the emission, the ACL emission for the cells. And here we, our experimental were supported and were in line with the simulation, and mainly in this first paper, in this first studies, we can visualize the first 5 micrometer of the ACL line. After Neso Sojic has this nice idea to use a permeabilization approach, so to permeabilize the cell's membrane and allow the TPA to reach the electrode also under the cell membrane. In this way, we can have a real map of the first, as I showed you before, the first few micrometer of the cells, and we can avoid these holes in the cells visualization, and we can have homogeneous ACL emission that is the emission only of the basal plane of the cells. This is something very similar to what is called near field microscopy, with a vertical resolution that is around 1 or 2 micrometer. So only the 1 or 2 micrometer up on the electrode surface called the mid. So you can have a vertical resolution from the electrode surface towards the bulk of the solution. That is quite interesting. And next step in this field of application, I think I'm fully convinced about that we need to find a way for control the emitting layer. So this 1 or 2 micrometer, we need to find a way for control this active area, active zone, in this example. In this example, we just published a paper where we demonstrate that with a chemical lens we can control the emitting layer. We can, by adding, in this case, by controlling the phosphate buffer concentration, we can have the emission. We can decrease or increase the emission layer, so the emission ACL active layer of 1 or 2 micrometer, depends from the PB concentration, phosphate buffer concentration. This is very interesting and will, I think, in the next future bring us to what I would like to call something similar to confocal electrochemiluminescence. That is my dream. Okay, let's move to the last part of this story, and is the combination that is on the combination between ACL, what is called remote ACL, so only TPA oxidation occurs, and the combination of ACL and dye-doped silica nanoparticles. We are doing this research activity in a strong collaboration with Luca Prodi and Enrico Rampazzo. The idea is very easy. At the beginning, 10 years ago, was very easy. So you have a ruthenium trace bipyridine, a single luminophore, and you have a nanoparticle. So you have more dye and you increase the signal. That's not the case of ACL, but the idea at the very beginning was very easy. There are other people that are combining ACL and nanotechnology. Here, there are my favorite to review. So this and this, by using a different nanomaterials. In our research activity, we have focused on the dye-doped silica nanoparticles, as I told you, in collaboration with Luca and Enrico, the dye-doped silica nanoparticle. So we started this story by with an approach of nanoparticle synthesis that was very novel, I think, at that time. And this approach of this synthetic approach is micelles induced synthesis of nanoparticles. So basically we use pluronic surfactant. Pluronic surfactant can generate micelle in water solution, and you have this micelle, and you use this micelle as a template for the synthesis of the nanoparticle. The advantage of the micelles, of course, it's obvious, but micelles have a hydrophilic shell and hydrophobic core. Okay, nice. So you can use this hydrophobic core for embed not soluble dye, not soluble in water. For example, diphenylanthracene or some molecules that are fully not soluble in water. And you can use this hydrophobic core for embed the hydrophobic molecules. And after that, you can grow the silica core, within the center of the micelles, and you can have a very nice and stable nanoparticles with the hydrophobic dye in the middle, and with hydrophobic dye in the middle, and this allows you to use hydrophobic dye as ACL emitter in water. That is one of the very important points. So the nanoparticle that they prepare is quite good and reproducible. So we have a silica core of 10 nanometers and a PEG shell, so a shell of 25 nanometers. So since we have these nanoparticles, we can put in the nanoparticle this different dye. This is work in the this is a publication in 2012, where we put in the core diphenylanthracene and rhodamine or cysteine, and we can have ACL threads. So we can tune the color of the emission by putting in the core different dye. But more interesting, I think, is we can use this synthetic approach to put in the nanoparticle, to add in the nanoparticle different amount of ruthenium. So we can exactly prepare nanoparticle with the same shape, with 2, 4, 6, and increasing the ruthenium concentration. And this is amazing because we can use the same molecule, the same nanomaterial, and see how it's changed the emission from 2 dyes to 24 dyes. And this is what we did. So here you can see ACL emission as a function of dye doping, and you see an increase of the linear. So of course, it seems obvious. You put more dye and the ACL increase, and we have this linear increase. But after, this is not fully true, because when we increase too much, when we go to very good doping level, so when we go to 20, 25 dye for each nanoparticle, we have a sort of plateau in the ACL. This means that there are other mechanisms involved in the ACL generation. There are other processes involved in this, because there is not a linear increase as a function of the dye doping. And what are the other effect? We measure, for example, Z potential, that I think is quite interesting. And here I reporting the red dots are the Z potential. You see that Z potential become almost positive at such in such doping level. So it means there are some charge involved. Nanoparticle become positively charged, even positively charged, and there are some electrostatic effects that are involved in the ACL generation. In fact, we are using this as with the co-reactant mechanism. With the co-reactant mechanism, so TPA radical cation cannot penetrate where the nanoparticles are positively charged into the shell of the nanoparticle. Okay, but why we go to such positive potential? Why we go to such a positive Z potential? Because ruthenium-3 bipyridine has 2 charge plus. So let's move from ruthenium-3 bipyridine to another inorganic complex that is a neutral one. So this is batofen complex. This is a neutral. And in fact, if we increase the doping level, we have almost still negative Z potential, and we are still negative Z potential. In fact, if we go to our neutral complex and measure the ACL emission as a function of the dye doping, we have, in this case, a linear increase of the ACL emission, because Z potential does not change too much, and we can have this linear increase in the ACL intensity. And by comparing the two dye-doped nanoparticles with the ruthenium batofen neutral dye or with the ruthenium-3 bipyridine, we have this increase, because in one case, the ruthenium-3 bipyridine, for this plateau due to the Z potential, while in the case of the neutral dye, we can have a linear trend. And this brings on my last slides, I think, on a new activity that we're doing, and the combination between dye-doped silica nanoparticles and magnetic beads. In fact, our final idea, our final proposal is always to go to the device, to the diagnosis. So we combine, in this very recent paper, we combine our nanoparticle, the nanoparticle produced by Luca Prodi and co-workers, our nanoparticle with magnetic beads. And this is electrode generation, so electrode oxidation, beads emission experiments, and you can see that we have around 8-fold of increase when we use nanoparticle instead than class standard ruthenium dye. This is a different synthesis, but I don't have time to go into detail on this. But what I have time, and I must go into details, is the stability. One of the main issue in ACL is the stability of the dye in the time. So when we have electrode, the beads on the electrode, we apply the potential, we typically observe, and here you can see from the movie, we typically observe, this is just a ruthenium one, we typically observe a decay of the emission and the deactivation of the dye. In the case of nanoparticle, again, here you see the movie on the nanoparticle, here you see the movie only related to ruthenium-3 bipyridine. And in the case of nanoparticle, the decay is much more slower compared with the standard dye. This is amazing, and I thank the reviewer, because we obtained this results by replying to the reviewer. So reviewers are always very important, and But we observed that the nanoparticle, and this is, I think, important message, nanoparticle protect against side reaction that occurs in the case of standard beads, protect against some side reaction, and we have a lower increase of the ACL intensity. I don't know how to go further. You can see the movie again. No. Okay, so just for conclude, I hope that I give you just very general conclusion. I hope that I give you the idea that ACL is a very powerful analytical technique for the visualization of real marker on a cell surface, or for the quantification of marker in a commercial application. The main message, except from our results, the main message is that we can increase the ACL signal, and this is, I think, is a very nice application. We can increase the ACL signal by fully understand the mechanism of the signal generation in the case of electrochemiluminescence, so in the case of co-reactants. So the full understanding of signal generation could give us an increase of the analytical signal. I think we like to call it amplification of the signal instead than amplification of the recognition of the biomolecules. So amplification of the signal thanks to the mechanism full understanding. So these are the people collaborating in this big ACL project, where this is our group, Professor Francesco Paolucci group, with the dyed-up silica nanoparticles. We are mainly do this in collaboration with Luca Prodi. We are doing some modeling and simulation, like I showed you, console simulation with the Professor Cristian Amatore. A single cell analysis were performed in collaboration with Neso Sojic, and we have this very nice carbon nanotubes Electrode with Andrea Slash and with Professor Maurizio Prato. This is the group in the last Zoom meeting, unfortunately. And I will leave you with this is last slide. ACL community is quite, I think, is a quite big community and very good community. We have a meeting each 2 years. The first one was organized in Bologna, in Bertinoro, very close to Bologna. The second one was organized by Professor Sojic in Bordeaux, and the third one were organized by Professor Gubauxu. It was very nice meeting with many people attending in Changchun. So in 2019, I hope that we will have next ACL workshop very soon, and not especially and not online. So with this, I would like to thank Michael again for this kind of invitation, and all of you for that kind attention.
Thanks so much, Giovanni. Brilliant. Like, as someone who's not an expert in ECL, it was very clear talk and very high impact work. Very impressive.
Thank you.